I'm going to discuss two subjects. I'm going to go through them really quickly, but I have a lot of slides with a lot of cool pictures. When we talk about rod, it's actually the umbrella that is two fungi. They're two new species, and they're actually two diseases, okay? I know that's kind of a complex type of thought, but when we first started working on it, we found Ceratocystis fimbriata, okay? Similar to other Ceratocystis fimbriata that occur on trees around the world. Well, we found a second Ceratocystis fimbriata, and that's when all our fun began, yeah? We called one Ceratocystis species A, one Ceratocystis species B, with a very large collaboration with scientists here and around the world. We now have named and described two new species of fungi, okay? Ceratocystis species A has become Ohia. B is Ohi'a. We now know they cause Ceratocystis wilt or Ohia wilt and Ohia canker. And for what to remember with these two species, the first one, Ohia, is actually more aggressive and more widespread. Okay, our paper is in press. I'm going to go through a lot of cool pictures really quickly, but for Ohia, you see a dead tree, you look inside the wood, you see very defined dark staining, and this is what these fungi look like growing on a plate, which unfortunately, that's what you're looking at down here, okay? When you look under a microscope, and now you start taking the mug shot of these fungi for Ohia, what's important to note is the types of spores being produced. So the fungus grows kind of like a spider web, and then it produces propagules, little babies, to start new infections. And they can be hat-shaped, they can be kind of cylindrical, and they can be round and dark. And depending on the types of spores being produced, some are short-lived, some are long-lived. And what that means on how the fungus can spread around and how new infections can start, it's very important to understand this aspect of the organisms, okay? So Ohia, Ohi'a, same dead tree, but when you look inside the wood, the staining's very light, faint. The patterns are different. Sometimes you see a dark line associated with it. And growing on the plate, really what you notice, the necks of those bottom right structures are shorter. Okay, now we know we're working with two different things. Okay. Just like humans, we like to do the family tree, the 23andMe. That's what you're looking at. You can't read it. But the purple is Ohia in relation to other Ceratocystis and Ceratocystis fimbriata around the world. And the green is actually Ohia. And they're separated into two big groups. You can ask me about stuff later. I could talk forever. But what's the take-home message? We're trying to figure out what species, our new species, are most genetically related to. And oftentimes this will give you a big clue on the types of hosts, like Ohia or other crops it would infect, and where they're located around the world. Okay, with all of that work being done, this is what we can say. There are two new species of Ceratocystis, okay? These two new species are only found on Hawaii Island. After all that work, we're still not sure how they got here. We're still not sure where exactly they're from. But the map indicates what region of the world the most similar cousins are located, okay? The Caribbean, Latin American region of species A, or Ohia, the Asian area, species B, Ohi'a. Now, this is where I told you it got complicated. We found Ceratocystis fimbriata, we found another Ceratocystis fimbriata. There are actually lots of Ceratocystis fimbriata already in Hawaii. You can see confusion. But we do know nothing that was already here evolved to these new pathogens, okay? So that's the message for a tale of two species. Complicated, but I think it'll become more clear as the day goes on. Okay, so we've learned a lot about the fungus. We're really interested in Ohi'a, and was every tree going to die? I mean, that was really the big concern when these diseases were found. So we've done a lot of work looking for host resistance in Metrosideros polymorpha varieties. Blaine got his master's degree in my lab actually studying the 4 main varieties that exist on Hawaii Island. I'm so sorry, I hear like...
It looks like it's rubbing. Is it hanging over there?
Sorry, it's my exciting, you know. I'll try to make it and not move around. But what he did was he took the 4 varieties, he took the more aggressive pathogen to start because we just didn't have enough seedlings to work with overall. So he inoculated it with Ceratocystis Ohi'a, and go through, you wait and you watch and you take measurements. So you try to figure out, well, how long does it take to actually wilt? You start to see those first symptoms. Do the seedlings die and how long does that take? You now actually look inside to where the fungus colonized, how much is discolored inside. You can calculate disease severity based on height of seedling and how much discoloration is present, okay? And we're looking really for resistance, something that actually just excludes or overcomes the fungus. The fungus can try to attack, but true resistance, the plant goes on happily living with almost like the fungus wasn't there, okay? But tolerance is also important too. So you can have fungal attack, but the tree actually doesn't die or suffers symptoms far less, okay? So both were important. And just to show you, yes, scientists got to give you graphs, but I'm going to summarize it really quickly. When you look inside, trying to figure out this disease severity, those 4 columns are actually the 4 varieties, okay? Glabrema, Newellii, Incana, and polymorpha. And what we're trying to measure is, compared to the height, how much discoloration did we find? Now, right away, you can see there are differences, okay? Now, we can also look at, well, how many of them died out of the total, okay? That's the blue columns. And if they died, how long did they take? Okay, and that's the yellow on the right column. So just to summarize, it was good news. Under high disease pressure, first of all, the varieties responded differently, okay, to the fungal attack. And actually, some seedlings have survived. And we're talking almost a year. We're letting them, you know, we didn't kill them. We want to see what's going to happen. Okay? So not only do we work with the varieties on Hawaii Island, but we're looking at the species that exist in the state and also throughout the Pacific region.